I'm Zhang, a PhD student in the Biotic Interaction Group of the Institute of Plant Sciences in University of Bern from Switzerland. I'm very glad that I have the opportunity to share my PhD project with you today. And the topic of my talk today is the evolution of plant defense resistance in natural enemies. And if you would like to see my abstract, it's in the book at pages 131 for the poster 85. So as we know, plants are facing many attacks from the herbivores. And as Anna said before, why the plants are still staying green? Two of the most important strategies we think for plant defenses is their direct and indirect defenses. For the direct defenses, the plants can producing many kinds of plant secondary metabolites that can be toxic and repellent to the herbivores, which can reduce their damage of the plants. And during the attack of the herbivores, the plants can also attract the natural enemies of the herbivores. So it is the indirect defense strategies. And by attract the natural enemies, the enemies can help the plant to kill or reduce the damage of the herbivores. And today, I'd like to tell you the interactions of the maize most important secondary metabolites, benzoxinoids, and the root herbivores and their natural enemies, the entomopogenic nematodes. So although the maize plants can release a high amount of benzoxinoids in the soil, they're still facing two important root herbivores. One is the banded cucumber beetles. They are generalist, and the other is a western cone rootworm, is a specialist. Especially for the western cone rootworms, it's originally coming from Mexico and widespread in the whole US. And now it's gradually invading to the Europe. Both of them, the larvae feeding on the root parts, and the adults are damaged on the above ground part. They're eating the leaves and also the fruits. And the most damage caused by the specialist western cone rootworms, it can cause a lot of damage of the root, and so then cause the falling down of the above ground part. So considering the major damage of the western cone rootworm, so it's also called the billion-dollar bugs. So the people now are struggling to find strategies to control the western cone rootworms, but basically still relying on the traditional control strategies, like the insecticides, Bt transgenic plants, and crop rotation with the soybeans. However, there are many literatures have reported that the western cone rootworm that gradually increasing the resistance to these three strategies. So now the farmers are looking to the biocontrol strategies by using entomopogenic nematodes. Now I'd like to shortly introduce the infection process of the natural enemy, the entomopogenic nematodes. When the young infective juveniles hatch from the host, they are trying to find a new host. When they locate the host, they're trying to penetrate into the host through the openings, and they will develop in several generations in their host, and then the larvae will gradually turn into orange and red. And after using all the nutrients of the host, they will hatch out from that and finding another host. And we already know they have a wide range of hosts, and they can gradually, widely used for the biocontrol strategies. And now we already know some farmers already start to using that by spraying that in the field to control the below ground pests. However, based on our previous studies, Krista already found that the western cone rootworms can sequester the benzoxinoids in their body and to fight against the entomopogenic nematodes. Basically, they have two strategies. First, the larvae can sequester the benzoxinoid in their body and transfer to amyl glucose. And here we could see that the nematodes are quite repelled by the amyl glucose. Another way is they can sequester benzoxinoids in their body as H-dimer glucose and also the breakdown products like amyl. And we found both H-dimbo-glucose and AMBO, they are very toxic to the nematodes. And to test how this influences their final efficiency, we reared the Western corn rootworm larvae in the two genotypes of the maize plants. One is a BX1 mutant and the other is a wild type. And then we analyzed the benzoxenoid concentrations in these two kinds of larvae. Here we can see the Western corn rootworm from feeding on the mutant plants, they have much lower benzoxenoids in their body. And then we use these two kinds of Western corn rootworms to do the nematode infectivity bioassays. And here we could see when the nematode infects the larvae feeding on the BX1 mutant plants, the infectivity indeed very higher than the others. And then we changed the background of the maize plants, and we have seen the similar pattern of the other. So from here, we can conclude that the Western corn rootworm sequestered benzoxenoids can indeed reduce the infectivity efficiency of the nematodes. However, all these bioassays are conducted by using a commercial strain of the nematodes, which has never contacted with Western corn rootworms before. So now we are looking back to the Western corn rootworm distribution in the world. As I mentioned before, it's original from Mexico and it spread all over the US. So based on this distribution, we can have the hypothesis that maybe during a long history of co-occurrence between the nematodes and the Western corn rootworms, the nematode can gradually increase their resistance to the benzoxenoids sequestered by the rootworms. So the main aim of my PhD project is to find out whether and how the nematodes can be resistant to the Western corn rootworm sequestered benzoxenoids. So based on this aim, we first worldwide collect 25 populations of the nematodes. Among that, 8 is from the US, which is originally with the rootworms, and the others have never contacted with the rootworms before. And here I have to say thank you to all my collaborators nicely to send us populations of the nematodes. And firstly, we identified the nematode species of these 25 populations. We found all of them are heterobacterous, and most of them are heterobacterous bacteriophora. The first step for this project is we conducted bioassays of the infectivity again in the BX1 mutant and wild-type maize-feeding rootworms. Here we could see a clear pattern that the nematodes from region without rootworms, they still have lower infectivity in the wild-type feeding rootworms, while the nematodes from region with rootworms, they are not influenced by the benzoxenoids in the larvae anymore. And to explain why it happened, I screened a set of factors that are related to the infectivity efficiency. And here I just show the 3 most important ones. Yes. The first one is the AMBO-glucose repellent effects on the nematodes that I conducted a petri dish experiment. And here we can see the clear trend that the nematodes from the region outside, like the vociferous distribution, we can see the nematodes are quite repellent by the AMBO-glucose. But the nematodes from the region with rootworms have a long history. They are not care about AMBO-glucose anymore. And then I tested the H-dimbo-glucose toxic effects on the nematodes. Here again, we see the nematodes collected from the region without rootworms. They are highly influenced by the H-dimbo-glucose. But the nematodes from the region with Western corn rootworms with a long history, they don't care about that. And then I tested the toxic effects of AMBO again. And here we don't see significant results, but indeed the trend is quite similar as H-dimbo-glucose. So from here we can conclude that the nematodes indeed increase their resistance to benzoxenoids sequestered by the Western corn rootworms during a long co-occurrence history with the corn rootworms. And to confirm the pattern I showed just now is indeed because the high long history of co-occurrence. We select one freshly collected nematode population, which are susceptible to benzoxinoids. We conduct the manual selection experiment. Here is a process for the manual selection experiment. Here we use RW14 nematodes, and we divide them into 20 batches. 10 of them is reared in the western corn rootworms, and the other 10 is reared in the bounded cucumber beetles. We also rear them in bounded cucumber beetle because they have very low concentration of benzoxinoids in their body, so here is like an active control. And we continue to rear the nematodes in these two kinds of insects for 5 generations, and after 5 generations of rearing, we did the infectivity test and phenotyping. Firstly, we tested how the infectivity efficiency changed after the first generation of rearing. And here we can clearly see only 1 generation of rearing in both western corn rootworms and the bounded cucumber beetles. They are still influenced a lot by the benzoxinoids in the larvae. And after 5 generations, we tested the infectivity of the nematodes on the western corn rootworms that feeding on the BX1 mutant and wild-type maize plants again. And here we can see clearly that the nematodes from the bounded cucumber beetles, they are still influenced a lot by the benzoxinoids in the larvae of the western corn rootworm. But after 5 generations rearing in western corn rootworms, we can see the infectivity even higher in the nematodes. They are infested on the wild-type plants. So we can see that after 5 generations, the nematodes already increase their efficiency by rearing the western corn rootworms. And to explain why it happened, I conducted the choice experiment again to see how the amber glucose will repel to the nematodes. And here we can see that the nematodes from bounded cucumber beetles, they are still repelled a lot by the amber glucose. But the nematodes from western corn rootworms, they are even attracted by the amber glucose. And again, for the H-dimble glucose and amber toxic effects, we can see that both of the compounds, they can still influence the nematodes from bounded cucumber beetles. And the nematodes reared from western corn rootworms, they can be resistant to both compounds. So from here, we can make conclusions like the entomopathogens nematode resistance to benzoxinoids is correlated with the length of evolutionary history with western corn rootworms. And the nematodes show a very fast adaptation to benzoxinoids sequestered by western corn rootworms. And the nematode resistant to benzoxinoid repellency and toxicity effects are the key factors for their adaptations. So in a word, plant toxin resistance is a novel breeding target for improving the biocontrol agent efficiency towards specialized insect pests. And for now, if you are still hungry about the interactions between the plants, root herbivores, and also the natural enemies, we are very welcome you to the poster 17 and 85. We could tell you more about the mating stories in the below ground. Finally, I indeed want to say thank you to the new Phytologist committee giving me the opportunity to give this talk, and also my supervisors and colleagues helped me to finish my PhD project, and also our collaborators from Missouri State University and National University. And thank you for your attention.